I'm going to get into our notation system now, and as you can see, there's colour. And what we have done here is essentially define. So this was the notation system back in 2010. We published a paper on this, and after this I'll send you this paper so you can have a look at it a bit more. But say what we've been doing over the years is working with people to model pathways, and of course when you discover a problem you can go, okay, well I can change it. And so that's what we've been doing. So I think the current version of the notation system is like this. And there's been I suppose an important number of changes since we originally described this system. We're just about to submit 2 papers on it, hopefully. It's me holding it up at the moment. And this is the scheme that we've developed for describing pathways. So we have this idea of entities. Okay, so entities are physical things. They are proteins, they are biochemicals, they are genes, etc. And likewise to the SBGN, we've selected a set of standard shapes in this case to depict these different entities. And we reinforce what they might be using colour. Now the colour doesn't, so a trapezoid like this says a drug, we always draw it yellow, but the yellow isn't important, it's the fact it's a trapezoid that really defines it as being a drug. And so a protein complex is just a rounded rectangle. The colon there is what actually the software uses to recognise the fact it's got more than one protein in it. And as I say, there's a range of shapes here that define different types of entities that we might want to represent. And unlike SBGN, we have an extensive palette of the nature of interaction. So rather than just drawing a little black square saying process node, we define all process nodes as binding, oligomerization, cleavage, autocleavage, catalysis. So these are all things that people have come to me while we're modeling pathways and said, you haven't got a process node that describes this process. We go, okay, we'll add one in. So all processes are circles, and that they have a 2 or 3 letter code to define what it is that they actually represent. So they describe not an entity, but a process that has occurred between entities that result in something downstream of that. Does that make sense? Then we have a simple set of lines. So for most interactions it's just an arrowhead straight like this. For catalytic reactions, and this again is taken from SBGN, we have this open round circle. We use a purple line here to reinforce it, but again it's the fact it's an open circle. In some of the things we've been looking at, we've been actually looking at interactions between neurons, and so we have an action potential as a type of thing that might happen. We also have this non-covalent interaction which is specific to us, and this means that actually when you're trying to draw a big protein complex, you've got, excuse me, that's my microphone. When you're trying to draw this big structure, biological structure, you want to be able to show things as being separate and not just in one big box, and we use that in there. And as we'll come on to, obviously inhibition is a very common concept in biology, that this inhibits that, and we have a number of arrows, and we'll come to this later. Now, as I say, the system was developed as a graphical notation system for drawing biological systems. What in later years, and since that first paper we've done, is we've changed that notation system now so that things which are so we now support essentially a computational approach for modeling pathways. So you can use your pathway to model the dynamics of a system using the diagram itself as the basis for that. And for this, we're going to talk about, in the second half of this, about Petri nets. And Petri nets are a formalized way that we can run simulations through diagrams. But Petri nets are composed of places. So all of your physical entities, if you like, are places. And all of your process nodes are transitions. Okay? And this won't mean anything to you now, but hopefully it'll make sense when we actually go on and talk about this. Now, the last part of this notation system is devoted to how you actually label things. As I say, one of the more frustrating things in biology is sloppy language, how you describe something. And so we always use standard gene notation. So even though that might not be what's used in the literature. So you can basically, the language that's used in literature is not always the language that we use in gene symbol notation. And so basically we're very stringent about how we actually label things. So if you have two proteins in a complex, you can actually put this colon, say there's protein name 1, protein name 2. And if there's more than one, we'll have this little bracket saying N, a number, so that says more than one of those. And we have this idea that a protein can be modified. So obviously proteins, the way that they often function is that they'll be phosphorylated, ubiquitinized, or one of those different processes. And that modification state is very important to understanding its biological activity. So we can support the idea, you can support that, and there's a whole range of those here. Okay, so we can get into that a little bit more, but we're very specific about how components are annotated or labelled, for a different word, and we can come on to that. So, basics of this. If you want to say that protein A translocates from the cytoplasm to the nucleus, you could draw this in this way. So A translocates, and though we would draw on the background of here this concept that on the left side we have the cytoplasm, on the right side that's the nucleus. We draw that transition zone at the nuclear membrane, and what we'd be showing is that this protein is translocating from one place to another. In this case, we're saying A and B bind to form AB. Okay? So we show, and one of the strict requirements of the process notation diagram is that you actually show the products of a reaction before you take it forward. You can't just draw two arrows saying A and B interact. A and B dissociates to form A and B. So here we're getting into what is, multiple reactions. So B catalyzes the phosphorylation of A. B and C, so this is Boolean logic, if you like, so that both of these guys are going to be required for that to happen. Okay, and this is the or statement where you can say B or C can catalyze that reaction. Does that make sense? So very simple, almost triplets of information that can be strung together to make sentences. I'll just make one very quick thing about the main thing here. We've tried where possible to use symbols that map onto the SBGN notation scheme. As I say, certain symbols such as this weirdness here, where you've got this two square corners and two rounded corners, we have actually just used it. So we use standard geometrical shapes for everything. So whereas perhaps SBGN might describe a receptor complex like this, we would just label it in the same way. So we're describing the same information as that we are seeing here. We're just using text-based version of it rather than trying to reinforce it. And so the argument might go, does this help you understand what that is better than that is? Is that better than that? I don't think so, really. And actually to construct that takes quite a lot of time and effort, because you've got to get a square, put all the bits in, label them all up, whereas here you just type in the actual sample labels. Okay. Actually, this is not the case anymore. We've got rid of all of those, so that's outdated. But the main thing here, or one of the main things here is, while they have only 3 specific processes that they actually define, we define many more. Okay, so a lot of processes here, phosphorylation and ubiquitination, are not defined within the SBGN, and that kind of has an issue downstream. Right, I'm not going to go through this. There is rules. You've got to have rules if you're going to have order, and I'm not going to go through these rules, but there are many of them.